Thank you very much for the kind introduction, and I'm very excited to put the spotlight on Talaromycosis today. And I'm going to be talking about risk factors for this disease in Vietnam, which we explored through a multi-center case control study. I don't have any conflicts of interest to declare. So this is Talaromycosis. It's caused by Talaromyces marnifae, which is one of the thermally dimorphic fungi. That means that it switches from a mold in the environment. It's inhaled by humans and at body temperature switches to this pathogenic yeast form, which survives in our macrophages and disseminates throughout the reticular endothelial system to cause a severe multi-system infection in immunocompromised people, mainly those with advanced HIV disease. Talaromyces marnifae is endemic to Southeast Asia, southern China and northeastern India. And in these dark purple regions here, it's actually a leading opportunistic infection, more common than cryptococcal meningitis and causing more deaths in these areas. But despite the very high disease burden and the significant mortality rate, this is a neglected disease and it's received little programmatic attention and resources. The ecology of Talaromycosis, I think, is a really fascinating area, but again, it's under-researched. What we've known for several decades is that the bamboo rat is the zoonotic reservoir for disease. Bamboo rats are these very large rodents. They can grow up to 0.5 meter long, which I think is a terrifying thought. And they are regarded as agricultural pests, but they're also farmed and eaten for meat in some areas. So environmental surveys have found the prevalence of Talaromyces marnifae among bamboo rats, all 4 species of bamboo rats, ranging between 10 to 90%. And they appear to carry this fungus asymptomatically. In Vietnam, what's interesting is the cases of human Talaromycosis appear to follow the distribution of the bamboo rat. Before our work, there had only been a single case control study performed way back in the 90s in Thailand, Chiang Mai, which is a mountainous region. You can see on the picture here, it's absolutely beautiful. What they found is no evidence of direct bamboo rat to human transmission. Rather, the risk factors for developing disease in Thailand were agricultural work and exposure to soil. Environmental studies have also found that there's a seasonality to Talaromycosis. So in the tropical monsoon season, the increased temperature, the rainfall, and the humidity appear to be favorable conditions for Talaromyces marnifae, and the incidence of human cases rises between 30 and 70%. We were interested in finding out the specific risk factors in Vietnam. Vietnam is unique because Talaromycosis occurs across a very diverse climatic and geographical range in both urban and rural environments. So we conducted a multi-center matched case control study of people living with advanced HIV disease. We collected data on 13 predefined exposure variables collected by face-to-face interview, and we performed a multivariable analysis to identify the independent risk factors for disease. Next, we did geographical mapping of cases and control, and finally, a post hoc telephone interview to assess previous travel or residence in highland regions, which we'd identified as potential hotspots for disease based on the distribution of bamboo rats and also the incidence of human cases. We recruited people from 2 major centres in the north and south of Vietnam that receive about 40% of all HIV referrals in the country. So let's talk about our results. We recruited 600 patients, 200 cases, over 400 controls. They were similar in terms of their age and sex. CD4 count was lower among the cases compared to the controls, but you can see that both were profoundly immunocompromised. There we go. And I think reiterating this is that the controls were diagnosed with a broad range of other opportunistic infections. So we had a very clinically relevant control group. In our multivariable analysis, the independent risk factors for disease were exposure to tropical plants. That includes rice, bamboo, and sugarcane, and also exposure to farmed animals. And these findings are in line with the previous work that was done nearly 30 years ago in Thailand. But where our two studies differed is that exposure to soil was not a risk factor for disease in our work. And what we hypothesized is that it's probably exposure to a very specific type of soil, which is associated with bamboo rats, animal excreta, and decaying tropical plant substrate. We think this type of soil is preferred by Talaromyces marnifae, and that disturbance of this soil through agricultural activities or through those extreme weather conditions in the monsoon season leads to aerosolization of the Talaromyces marnifae and increases the respiratory exposure in humans. Geographical mapping was also very informative. So what we found is that patients in the highland regions were 9 to 11 times more likely to develop disease than those residing in the lowlands of the Mekong Delta, and 3 times more likely than those in the Ho Chi Minh City area. But what was really interesting is that we found patients with previous residence or travel to highland regions at any point in their life were at 3 times increased risk of developing Talaromycosis. And that brought about the idea, our study was able to demonstrate this epidemiological link between human Talaromycosis and residence or travel to highland regions, the hotspots. And our findings offer potential insights into disease latency and the natural history of Talaromycosis. We know that Talaromycosis can occur as a latent infection because there have been case studies of travelers who have visited endemic regions years before that later developed Talaromycosis. And I think that really adds to the evidence of this being a latent disease. Also, our data can inform prevention strategies, including patient education and the consideration of primary itraconazole prophylaxis for high-risk individuals with advanced HIV disease who are considering travel or moving to regional hotspots. There were a few limitations to our study, so a small risk of selection bias in 31 participants who were screened but not ultimately recruited due to discharge or death. The reference standard of culture is suboptimal in terms of its sensitivity. So there was a potential for misclassification of controls that maybe should have been cases. And finally, there was an imbalance in cases from Ho Chi Minh versus Hanoi due to earlier recruitment in the Ho Chi Minh City area, so some slight overrepresentation of people in southern Vietnam. In terms of future directions, well, we advocate for further studies to uncover more specific hotspots within those highland areas and identify more risk behaviours. We also advocate for further studies to define disease latency and the natural history of Talaromycosis. And I'm really excited to share with you some of the work of the Tropical Medicine Research Centre for Talaromycosis in Ho Chi Minh City, which is led by Associate Professor Thuy Le. They're doing some fantastic work optimizing qPCR detection of Talaromyces marnifae in soil and air samples. And they're using that optimized method to really define the prevalence in soil and air samples across a very diverse range of environments and seasons to identify more specific hotspots for this pathogen. And they're also linking that with exposure in humans, looking at IgG prevalence. So some amazing work. Our study is going to be published in Emerging Infectious Diseases next month. I'd like to credit my co-author, Dr. Bryant Jonah, and of course, my mentor and supervisor, Associate Professor Thuy Le, who's taught me so much about Talaromycosis and also about research and life in general. So I'm really grateful to have the opportunity to speak to you today and welcome any questions. Thank you.
Yeah.